Hello everyone and welcome to IVSA Bidar's official YouTube channel Pashuvani. Pashuvani is a platform that provides you with veterinary education. Our video series consists of practical relevant lecture videos, field approach clinical aspects, guide and tip for future careers, smart content related to both animal and veterinary science. We believe sharing is caring, so please like, comment and subscribe for further updates. And now, let's welcome a speaker of today's episode of Saturday's Sharing.
Hello everyone, I am Saikiran, currently a third year BVSc&AH student studying in Veterinary College Bidar. In this video, I am going to talk about epidemiology. This presentation includes the topic of epidemiology, few definitions and its objectives, determinants of a disease, and transmission and maintenance of infection. Epidemiology is derived from three words: epi meaning upon, demos meaning people, and logos meaning study. That is, epidemiology is the study of that which is upon the people. The other definition of epidemiology is it is the study of disease in a population and of the factors that determine its occurrence. That is, the factors which influence or facilitate the occurrence of a disease. Epidemics is the outbreak of disease in human population. Epizootics is the outbreak of disease in animal population, and apoarnetics is the outbreak of disease in avian population. There are 5 objectives of epidemiology. First is determining the origin of a disease whose cause is known. That is, when a disease occurs whose cause is known, it is important to determine its origin for the control of the disease. Second is investigating and controlling a disease whose cause is either unknown or poorly understood. That is, when a disease occurs whose cause is not properly known, then epidemiological investigations are required for its control. Third is collecting information on the ecology and natural history of the disease. This is required to predict where and when a disease might occur. Fourth is planning, monitoring and assessment of disease control programs. Disease control programs require proper planning and also monitoring and surveillance of the program. Fifth is assessment of economic effects of a disease and its control. That is to see whether the control program is economically beneficial or a loss. Determinants of a disease. A determinant is any character that affects the health of a population. Determinant is the main cause of an infection or disease. Therefore, knowledge of determinants is required for proper disease prevention. There are 3 types of determinants. First is primary and secondary, next is intrinsic and extrinsic, and the third is determinants associated with host, agent or environment. First is the primary and secondary determinants. The primary determinants are the factors whose variations have a major effect in inducing an infection or a disease. That is, these are the determinants which are main cause or the main reason for occurrence of a disease. And these are sometimes the necessary causes, like in the exposure to distemper virus is the primary determinant, which is also the main reason for the canine distemper. Secondary determinants correspond to predisposing, enabling and reinforcing factors. Like, these are the determinants which indirectly influence or facilitate the occurrence of a disease. Like, the season is secondary determinant in clostridial infection, and notably the monsoon season helps in the widespread of clostridial infections. Next is intrinsic and extrinsic determinants. Intrinsic determinants are those which are internal to the host, and also these are known as endogenous determinants, like the genes, the species, breed, sex, age. These are all the intrinsic determinants. Extrinsic determinants are those which are external to the host, like transportation, which can be a determinant in the causing of bruising in the carcasses, and these are also known as exogenous determinants. Next are the determinants associated with host, agent and environment. Sometimes these 3 factors are collectively known as the triad. The simple diseases are diseases in which only agent is the main determinant and host and environment are not much of a importance, like in the foot and mouth disease. And complex diseases are those in which all the three factors, the host, agent and environment, are involved, like in the mastitis, in which the host, which are normally susceptible in early lactation period, and the agent which causes mastitis, and also the environment, which can be the poor environmental hygiene. All these factors are required for the occurrence of mastitis. First are the host determinants and genotype. It is the genetic constitution of a host, and alteration in these genes can cause genetic diseases, which can also be inherited by the succeeding generations. There are three categories of genetic diseases. First is chromosomal disorders, next is Mendelian disorders, and then multifactorial disorders. The first two are purely completely based on the genes or the chromosomes, and the third is based on multiple factors like the name indicates. That is, there are involvement of multiple factors for the occurrence of a disease, like in canine hip dysplasia, where it requires both genetic weakness of the muscles and also the conditions which favor separation of the joint for the canine hip dysplasia to occur. Next is the age determinant. Age can also be a determinant in occurrence of many diseases, like the bacterial and viral diseases can be mostly seen in young animals than in the old animals. This can be because of the absence of acquired immunity or low host resistance of the young animals. But in contrast, the protozoan and rickettsial diseases occur more in old animals than in the young animals. Next is the sex determinants. There are four determinants in this. First is the hormonal determinant. Sex hormones can also affect the cause of disease in an animal. Like diabetes mellitus can be seen more in bitches than in the dogs because of the increased insulin requirements during the estrus. And the mammary carcinoma is less in neutralized bitches than in the normal bitches because of the effect of estrogen on the tumor. Next, there is occupational determinant. Sex associated occupational hazards can also be seen in animals, like a heartworm infection in male dogs is common than in the bitches because male dogs are often taken out as a hunting animal, therefore it can be seen more in the male dogs. Next is the social and ethological determinants. The social behavior of animals can also aid in the occurrence of diseases, like competition between male dogs for breeding with females or the territory can assist in the transmission of rabies through the bite wounds in a fight, or the transmission of tuberculosis from opossum to cattle. The opossums, when they are confronted by the cattle, they don't run away, they stand their ground. Therefore, there is a higher chance of transmission of the aerosols, and there is transmission of tuberculosis between the opossum and cattle. But the beagers, uh, their first response is to run away when they are threatened. Therefore, there is less chance of transmission of disease between them and the cattle. Next is the genetic determinants. Genetic differences in disease may be because of the sex linked or sex limited or sex influenced diseases. Sex linked are those which can normally be called as the Mendelian diseases, that is which occur because of the genes X and Y chromosomes and all, like the canine hemophilia A and B. Sex limited diseases are those which are limited to a particular sex and cannot be seen in the other sex, like cryptorchidism, which can be seen in only the male animals. Or sex influenced are those which can be seen more in one particular sex than in the other sex. Next is the species and breed. Both species and breed have certain effects, and also they vary in their susceptibility and response to different infectious agents. Like the species of cattle are more susceptible to airborne infections than the species of pigs, like in foot and mouth disease, which is more prevalent in cattle than in the pigs. and the species of dogs do not develop heart water, and the breeds such as Rottweiler and Doberman, they react more to canine parvovirus enteritis, and the breeds of boxers, they are more susceptible to mycotic diseases than the other breeds. This can be because the presence or absence of specific receptors for the infectious agents. If there is no receptor present in the host, then the infectious agent cannot express themselves, and there won't be any disease. Like poliovirus in monkeys do not have any specific or the proper receptors for the poliovirus, therefore they do not contract poliovirus. Next is the size and conformation. Size can also be a determinant, like the hip dysplasia or osteosarcoma can be seen more in the large breeds than in the small breeds of dogs, and conformation may increase the risk of some diseases in animals, like the cows which have smaller pelvic outlet compared to their body are usually predisposed to dystocia. Next is the coat color. Coat color can also influence some diseases in few animals. Like in white cats, there is higher incidence of cutaneous squamous cell carcinoma. This is because the white cats they lack pigmentation, which protect them from the carcinogenic effects of the UV lights. Therefore, they are at a higher risk to the cutaneous squamous cell carcinoma. Next, physiological status. Milk fever can occur in cattle which have just calved, because there is a high physiological stress on them. Agent determinants. Ability of an agent to infect is related to the susceptibility and immunity of the host. That is, if the susceptibility is high and the immunity is low, then there is a high chance of infection. Virulence is the ability of an infectious agent to cause disease in a particular host, and this may be in regard to the severity of the infection. It is the ratio of number of clinical cases to the number of animals infected. Next is pathogenicity. It is the quality of disease induction. It is also the ratio number of individuals developing clinical illness to the number of individuals that are exposed to the infection. A highly pathogenic organism induces disease in large portion of the population. That is, if an organism is said to be highly pathogenic, then it means that it can cause disease or infection in larger portions of the population. Next are the environmental determinants. First is location. The local geological formations, vegetation, and climate can affect the distribution of both the animals and the diseases which are related to it. Example is the chronic canine pulmonary disease, which can be seen in dogs that are usually present in the urban residents. This is because there is high density of dogs in urban areas, and also there is higher environmental pollution which can facilitate the spread of the disease. Next is climate. Climate, it has two types. First is macroclimate and microclimate. The macroclimate consists of normal components of weather, like the temperature, rainfall, wind, etc., to which the animals are exposed. Like the low temperature can induce hypothermia, which is more common in the newborns, and the wind and rain can increase heat loss from the body of the animal. Cold stress can predispose animal to disease. Wind can carry infectious agents like the foot and mouth disease. Solar radiation induces carcinoma, like the example we saw in the white cats, and humidity can also help in the survival of few infectious agents. The macroclimate can determine the vegetation, and it affects the distribution of disease, because the distribution of vegetation can also result in the distribution of hosts and its vectors. And Macroclimatic changes can alter the frequency and also distribution of disease. Next is microclimate. It is the climate that occurs in a small defined space, and it can range from few millimeters to a large area. Terrestrial microclimate is the microclimate that occurs over the surface of the leaves, and this affects the development of arthropods and helminths. The biological microclimate is the microclimate that occurs over the surface of host's body, and changes in this microclimate can change during the course of a disease, and it assists in the spread of the disease. Like during the parasitemic phase of human malaria, there is increased humidity of the body surface, and because of this, more mosquitoes are attracted to the body, and therefore there is a higher chance of spread of the disease. Next is husbandry. In that, first is housing. The housing can also predispose the animal to diseases, like the structure of the bedding materials and also the surface. Claw lesions can be seen in pigs more commonly, which are reared on aluminum slats than on the steel or concrete slabs. And if there is excessive floor slope, then this may also predispose the pigs to rectal prolapse. Next is the diet, which has effects in disease that are usually caused by deficiencies of protein, vitamins, energy, minerals, and all. Next is the management. If there is no proper management, then this can increase the spread of diseases. Like if there is increased densities of the population of the animals, then there is increased spread of the disease, because there will be an increased challenge of microbial pathogens. And also the use to which the animal is put can also affect the disease occurrence. Like hump sore is common in cattle, which are usually used for draught purposes, than in the cattle which are not used for heavy works. Next is the stress. Stress is the sum of biological reaction to any adverse physical, mental, and emotional stimulus that can disturb the homeostasis of the animal. And the factors which produce stress are known as stressors. Sally formulated a theory in which he described the effects of the stressors in laboratory animals, and these effects were combinedly known as the general adaptation syndrome, which were divided in three parts. The first phase was the general alarm reaction, which occurred after 6 to 48 hours of the exposure of a stressor. In this phase, there is a decrease in size of organs like the lymph, spleen, thymus, and also lymph nodes, and erosions could be seen in the digestive tract. The second phase is the phase of resistance, which occurs after 48 hours of the exposure, and in this there is adrenal enlargement, and also there is release of corticosteroids. And then there is a third phase, it is known as phase of reaction, in which death may also occur. Transmission and maintenance of infection. When a pathogen invades a host body, this causes infections. The survival of infectious agents can depend on their successful transmission to a susceptible host, initiation of the infection in the host, and also the replication of the agent. Transmission can take place in two ways: horizontal transmission or vertical transmission. First is the horizontal transmission. In this, infections are transmitted from one part of the population to the other part of population, like the transmission of an infection between horses in the same stable. In this, there are two types. First is direct transmission, in which a susceptible host can acquire an infection through the direct contact with an infected host or its infected discharges, like canine distemper, which can transmit through the infected urine or feces also. And second is the indirect transmission, in which an intermediate vehicle is involved for the transmission of the agent from infectious host to the susceptible host. Airborne transmissions are generally known as indirect transmission, but more correctly it is a direct transmission, because there is no involvement of any intermediate vehicle. Factors associated with the spread of infection. Characteristics of hosts. A host's susceptibility and infectiousness can determine its ability to transmit infection. Susceptibility to a particular infectious agent can be limited to a single species, or it can be in many different species, like equine rhino pneumonitis, which is limited to only single species of the equines, and the rabies, which can be seen in many different species. Infectiousness is the duration of period or the time period for which an animal was infective and the relative amount of an infectious agent that the infected animal can transmit. Diseases with short incubation periods, that is the period from the infection to the appearance of clinical signs, these run a clinical course that ends quickly either in recovery or death of the host, and for these kind of diseases high host density is required. Like distemper virus infection is endemic in urban areas where there is a high density of dogs. Next is the threshold level. This is the minimum concentration of an infectious agent that should be present in a host circulation for the transmission from host to a vector. And if this threshold level is not achieved, then there is no transmission of the infectious agent. Some animals can be infected, but they will not transmit the infection because the threshold level may not be reached. These type of hosts are known as dead end hosts. For example, Colombian ground squirrel is the dead end host for the Colorado tick fever, and the golden mantled ground squirrel can transmit the infection. In this graph, it can be seen that the Colombian ground squirrel, as the days passes, it does not achieve the threshold level, but the golden mantled ground squirrel can easily achieve the threshold level. Therefore, it can transmit the infection. Characteristics of pathogen. First is the infectivity. It is the amount of an organism that is required to initiate the infection in a host. Particle infectivity ratio. It gives an idea of number of particles or the organisms required to initiate the infection. For bacterial virus, that is the bacteriophages in tissue culture, it is 1:1. That is, one organism is enough to initiate the infection, and this is the high degree of infectivity. For animal virus, it is between 10:1 and 100:1, and for plant virus, it is 1000:1. That is, 1000 organisms are required to initiate the infection, and it is the lowest degree of infectivity. Infectivity can also depend on the route of infection, the age, and the resistance of host. Next is the virulence. It is the severity of infection and can be expressed by describing the morbidity and mortality of the infection. Morbidity is the number of animals infected, and mortality is the number of deaths because of the infection. If these are high, then it is said that the organism has higher virulence. Next is the stability. It is the length of time for which an organism can remain infective outside its host. Some organisms can survive for shorter period and are known as labile organisms, and others are resistant and can survive for longer periods. Stability is also facilitated with protective capsules such as the bacterial spores, which increase the stability of the organism. Affective contact. It describes the conditions under which infection is likely to occur, or the conditions which cause the infection. For a particular infection, it depends on the stability of the organism, route by which the organism leaves an infected host and enters a new susceptible host. It may be short, as in the seasonal diseases, or long, like the anthrax spores in soil, which usually are viable for longer periods. Behavior of an animal can change during the infection, and this can increase the likelihood of effective contact. That is, this can increase the chances of transmission of the infection. For example, the feral animals are naturally shy, but when they are infected with rabies, they start entering the houses, and this increases the chance of transmission of diseases from the feral animals to the human beings. And the stickleback fishes, when they are infected with cestodes, they become sluggish and their movement is quite slowed down, and this increases the chance of them being caught by the birds and increases the chance of transmission. Pathogenesis of disease may also increase the likelihood of transmission, like coughing and sneezing in respiratory diseases. This increases the chance of transmission by dispersing the organisms in the aerosols which infect the other hosts. Routes of infection. The site or sites by which an infectious agent enters and leaves a host is known as routes of infection. First is the oral route. It is one of the most commonest routes. Organisms like the rotavirus and salmonella contaminate water and foodstuff, and these act as fomites. Fomites are any inanimate objects that can transmit infection. When these fomites are ingested by the host, then the organisms also gain entry into the host body. These ingested agents can be excreted in feces, which produces the fecal oral transmission cycle. The agents can also be excreted through other routes like milk or urine discharge, as in Brucella abortus. Low pH of gastric secretion acts as a barrier against the organisms which gain entry through this route. Second is the respiratory route. It is also one of the commonest routes. Agents won't occur as a single organism, but they are usually associated with organic matter like droplets or dust, and these droplets or dust are inhaled by the host. And this is common in areas which have high density and poor ventilation. Third is the skin, cornea, and mucous membrane. Transmission via skin is known as percutaneous transmission, and it can occur through direct contact or a foam, and this is influenced by the population density and susceptible host. That is, if there is high population density and increased number of susceptible hosts, then the transmission of infection is more. Most of the time, the intact skin acts as a barrier for many agents, but some organisms can also penetrate this barrier and cause infection, like the blood flukes and hookworms. Infection by a variety of organisms occurs if the skin is cut or abraded, and this produces localized infection. That is, the infection is restricted to a particular area. Other agents enter percutaneously and produce generalized infections. Agents present in saliva, as in the case of rabies, can be transmitted through animal bites. The infection of cornea may be localized, like in the bovine keratoconjunctivitis, in which the infection is restricted to the eyes only, or it can spread to other parts, like in the Newcastle disease in birds. In this, the infection may start from the eye, but it can spread to other organs also. Some diseases can be transmitted through infecting the undamaged mucous membranes. Methods of transmission. First is ingestion, which can be through foams like contaminated water, or by the ingestion of intermediate host like the cystode cysts in the meat, which is consumed by the host. And these can be excreted through feces, urine, or breath. Next is the aerial transmission, which is the transmission of infectious agents through contaminated air which is inhaled by the host. And this is the common method of transmission of fungal spores and bacteria and pathogens which are related to respiratory tract. And direct infection from the expirated droplets is limited to the area which is directly in front of the infected individual. This is because the droplets are not small enough to be carried for longer distances. Therefore, they sediment as soon as they get out of the host body. Next is the contact transmission. This does not involve any external medium or transmission factor for its transmission, and this can be by touch, bites, or scratches. Next is inoculation, which is the introduction of infectious agents into the host body by puncturing the skin or through a wound, and it is associated with contact transmission like the bites from rabid dogs. The bite from rabid dogs can be both contact transmission and inoculation. It is spread through bites of the dogs, and therefore it is contact transmission, and it is indirectly puncturing the skin and inoculating the infectious agents into the body. Therefore, it is inoculation transmission. Next is the iatrogenic transmission, which literally means created by a doctor. That is, these are the infections which are transferred during surgical or medical practices by the doctor. And there are two types. First is the introduction of pathogen by contaminated or dirty instruments, and second is the introduction of pathogens which contaminate the therapeutic preparations like the drugs or the anesthesia which is injected into the host body, and this can also occur rarely by organ transplantation. Next is the coitus. These are the infections are transmitted through coitus, and these are known as venereal diseases or sexually transmitted diseases. And this is not just only limited to the vertebrates but can also occur in arthropods like African swine fever virus, which can be sexually transmitted from males to the female sticks of the genus Ornithodox. The mode of transmission frequently governs the epidemic picture, like the agents which are transmitted through the fecal oral and the airborne modes can produce sudden and explosive epidemics because there is a fast rate of transmission of infection, and coitally transmitted diseases spread more slowly because there is slower rate of transmission of the infection. This was about the horizontal transmission. Next is the vertical transmission, which is the transmission from one generation to the next generation. It has two types, hereditary and congenital. Hereditary disease are those which are carried in the genes of the parents and are transmitted to the offsprings, like in the retrovirus, which have their DNA copies in the host genome and is transmitted to the offsprings. The congenital disease are those which are present at the birth, and these do not involve any genes. These are acquired either in utero, that is inside the uterus, or in ovo rather than being inherited. And these may produce either abortion, teratoma, or innate infection. Innate infection are those which are inapparent and can continue even after birth. Germinative transmission is the infection of either superficial layers of the ovary or the ovum itself, and transmission to the embryo can occur through placenta or fetal circulation. But infection of the placenta does not always mean that the fetus is also infected. Next is the maintenance of infection. There are hazards for the infectious agents even if it is inside the host or outside the host. First is the environment within the host. Host has its own natural defense mechanisms like chemicals, acids, reactive cells, phagocytes, antibodies, which protect it from external agents. A successful parasite should be able to avoid all these defense mechanisms and also avoid competition with the agents which infect the host at the same time. Parasites have also evolved their own strategies against these mechanisms, like acid resistant helminth cuticle for the gastric acids and intracellular mode of life that protect it from antibodies, and the capsules which protect it from phagocytes. Next is the external environment. In the external environment, desiccation and UV light are the two main hazards. Desiccation may not always be lethal or fatal, but it frequently inhibits the multiplication of the agent. Agents can be protected from desiccation by being discharged in moist carriers like the feces and urine, or favorable surroundings like high moisture content, or in foams like the water and feed stuff. Maintenance strategies. These are the strategies adopted by the organisms to overcome the harshness of the environment. First is the avoidance of stage in the external environment. The idea behind this is that if the external environment is harsh, then it is better to completely avoid it. And it can be done by vertical transmission like the congenital or hereditary diseases, venereal transmission, which is the sexually transmitted diseases, vector transmission, and transmission by sacrophagia, which is the flesh eating, as in the case of Trichinella spiralis, which get transmitted by the flesh eating of the infected animal. Next is the development of resistant forms. In this, the infectious agents surround themselves with shells which are resistant to heat and desiccation, like the spores of bacteria and fungi and the cysts of helminth and protozoa, which protect them from the external environment. Rapidly in and rapidly out strategy. In this, the agents enter the host, they replicate, and they leave very quickly even before the host can launch an immune response against them. But this strategy requires a continuous supply of susceptible hosts because these agents have to regularly keep on changing hosts quickly. And next is the extension of host range. Many infectious agents can affect more than one host, and it is facilitated by the presence of different hosts in the same area. But this does not always means that there is a transfer between them. And some hosts can also act as dead end hosts, as in the case of elephant, which acts as dead end host for foot and mouth disease. Next is persistence within the host. The infectious agents can remain in the host's body for their lifetime. This is because the host defense mechanisms cannot completely remove them, and it can occur if the microorganisms adapt themselves to the host phagocytic cells, or they develop strategies to avoid the host immune response by immunosuppression and tolerance. Immunosuppression can lead to the agent being maintained in the host body for longer period, and it may be general or antigen specific. In general immunosuppression, there is survival of the affecting agent and also other agents, but antigen specific is one which involves response to only the infecting organism, but the response to the other agents is unaffected. That is, in this only the infecting organism can survive, but the other organisms cannot survive. Tolerance is due to the primary lack of responsiveness by the host. Persistence can be associated with long incubation period or prepatent period. Incubation period is from the infection till the appearance of clinical signs, and prepatent period is from infection till the shedding of the agent. Example is slow virus diseases like Medi Visna in shape. In this, the neurological and respiratory signs appear with an incubation period of 2 to 8 years. That is, it takes a long time to even show the clinical signs. Alternatively, an agent's prepatent period may be relatively short, but the excretion of the agent may continue for a long time, like leptospira infection in cattle, in which the bacteria is excreted in urine for about 1 to 2 years. Thank you.